Welcome again to the latest edition of the AJP-Heart and Circulation video table of contents, or From the Editor's Desk. In this issue, we've published 3 reviews, an editorial focus, and 15 original research articles. The issue is chock full of great novel and innovative cardiovascular science, so let's take a peek. Bowles et al. Reviews adaptive changes in the right ventricle in response to a variety of insults. This outstanding review covers integrative and cellular pathways related to right ventricular failure. The authors cover neurohumoral mechanisms, inflammation, and potential therapeutic options related to novel molecular targets. Chen et al. Set out to explore the role of capillary disorder in coronary ischemic congestive heart failure in rats subjected to aortic banding and ischemia reperfusion injury. Using a plastic fluorescent embedding technique, they found significant stenosis in all levels of the coronary arterial tree. Capillary density was increased in heart failure. They proposed that this disorder in capillary density and architecture may contribute to myocyte dysfunction in this model of chronic heart failure. Mura and colleagues subjected ENOS deficient mice to either 3-month ad lib feeding or calorie restriction. Isolated perfused hearts were subjected to 25 minutes of global ischemia followed by 60 minutes of reperfusion. The degree of myocardial ischemia reperfusion injury in ad libitum fed ENOS knockout mice was more severe than that in ad libitum fed wild type mice. Furthermore, calorie restriction did not exert cardioprotection in ENOS knockout mice. ENOS knockout mice exhibited elevated blood pressure and left ventricular hypertrophy compared with wild-type mice, although they underwent calorie restriction. Resveratrol enhanced cardiac SIRT1 activity but failed to mimic calorie restriction-induced cardioprotection in ENOS knockout mice. These results demonstrate the essential roles of ENOS in the development of calorie restriction-induced cardioprotection and SIRT1 activation during calorie restriction. Finally, Fukai et al. Demonstrate that proline-rich tyrosine kinase 2, a non-receptor type protein tyrosine kinase, plays a crucial role in the pathogenesis of pulmonary hypertension using an animal model of hypoxia-induced pulmonary hypertension. Resistance to a hypoxia-induced pulmonary hypertension was markedly higher in proline-rich tyrosine kinase deficient mice than in wild-type mice. An in vitro study using human pulmonary smooth muscle cells showed that hypoxic stress increased the proliferation and migration of cells in a PYK2-dependent manner. Proline tyrosine kinase 2 may represent a promising therapeutic target for pulmonary arterial hypertension in a clinical setting. Thank you once again for joining me for these video highlights. Please go to the AJP-Heart and Circulation website for more great cardiovascular science. We will see you in 2 weeks for more great science highlights. Don't forget to send us your best science now and look for our numerous calls for papers listed on the website. See you in 2 weeks.